Vivek sir, please begin.
Okay, sir. Thank you. So, good morning, all. I, Dr. Vivek Srivastav, Associate Professor, School of Pharmacy, welcomes you all to the webinar on Dendrimers in Drug Delivery and Targeting. Honorable Dr. M.N. Patel, sir, Provost, Parul University; Dr. Abhay Dharamsi, sir, Dean, Faculty of Pharmacy and Principal, Parul Institute of Pharmacy; Dr. G. Chakrabarty, Principal, Parul Institute of Pharmacy and Research; Dr. Lalita Jha, Madam, Principal, School of Pharmacy; and Dr. Umesh Gupta, sir, Assistant Professor, Department of Pharmacy, Central University of Rajasthan; and dear participants. So, due to the small size of the dendrimers, they are used in nanomedicine research. They are found to be useful as a delivery or carrier system for drugs and genes, but studies have shown that some dendrimers have medical use of their own, mostly due to their antifungal, antibacterial, and cytotoxic properties. And today we are fortunate to have Dr. Umesh Gupta sir with us to speak on dendrimers in drug delivery and targeting. Now, I will take a moment to introduce our speaker, Dr. Umesh Gupta, to you. Dr. Umesh Gupta sir is currently working as Assistant Professor at Department of Pharmacy, Central University of Rajasthan, India. He did his PhD in Pharmaceutical Sciences from Dr. Hari Singh Gaur University under the mentorship of Professor N.K. Jain sir. He has been recently awarded with AAPS Distinguished Young Educator and Researcher Award 2019 to attend annual conference of AAPS 2019 Farm Science 360 at San Antonio, Texas, USA. He is also recipient of first Chancellor's Award for Best Faculty 2018 at Central University of Rajasthan, Ajmer, India. He has been also awarded DAAD Research Stage for Academic and Scientist 2017 at Leibniz Institute for Polymer Fusion, Dresden, Germany. He has the past experience of working as a research scientist at Ranbaxy Research Laboratories, India, and postdoctoral research associateship at South Dakota State University, USA. He has more than 10 years of research and academic experience. The area of research is mainly focused on dendrimer-mediated drug delivery, nanotechnology, solubilization, and targeting. His academic attainments are many. He has more than 73 publications, 3 edited books, 17 book chapters, and 1 patent published to his credit, with a total citation of more than 5,420. The H indexed 34 as per Scopus and 35 as per Google Scholar. So, with this, not taking much of your time, I request Umesh Gupta sir to have his talk, sir. So, welcome, sir. I request you to present your lecture.
Thank you so much. Thank you so much for inviting me. Thank you, Vivek sir. First of all, I would like to thank all the faculty members, principal of the Parul Institute of Pharmacy and Parul Institute of Research and Technology for giving me an opportunity to share my views on the design topic on dendrimers. And it is a kind of privilege that I got this opportunity to present some of the views and basics about dendrimers in front of you. Thank you so much once again for inviting me. Now let me connect with my presentation. So give me a minute and I'll just start with my presentation. I hope all of you can see my screen, right?
Yes, sir. Yes, sir.
All right. So again, basically, this is something I would first like to connect the audience, because I know that there are many students who are who are from bachelor's degree, who are doing their bachelor's degree, and there are master's students also in the audience. For the bachelor's student, I will just say that this is a topic which is something like related to a new drug carrier system, which is going to deliver drugs only, but it is not tablet, it is not capsule, it is not syrup, it is not a liquid formulation. Rather, it is something which is having a very less size. In fact, it is the size less than 100 nanometer. So we are going to discuss a carrier system, basically a drug carrier system, whose name is Dendrimer. and we will try to understand what is dendrimer, right? What are the different types? What are the criticalities? What are the advantages? What are the disadvantages? How actually we can utilize this carrier in delivering drugs? Is it having some advantages even better than existing carriers, or it is something a very simplistic carrier, or whatever? So the topic is dendrimers in drug delivery and targeting. Those who are in the third year or seventh, eighth semester, they already know that what the targeting is, that we deliver the drug directly to the affected organ or to the affected tissue or to the area where the actual effect is there. So we call it as a targeting. In delivery, we know that tablet is also a drug delivery carrier system, right? It is a dosage form. It delivers drug. So dendrimers in drug delivery and targeting. Now, more you will study about dendrimer, you need to understand the concept of nano, the concept of nanotechnology, and you will realize and you will see that day by day, if you see that the carrier systems which are gaining momentum, or the drug carrier system which are having more and more attention, or you can say the research in the carriers which has resulted into an output which is very promising and advantageous, is nothing but the nano carriers. Now, what is basically nano? If you think about that, and if you remember just 10 to 15 years back, you will realize that the micro technology was taking momentum. We remember the micro tech, micro word, microfibers, right? That was taking too much momentum. But nowadays, if you see in the last 10 years, 15 years, you might have realized that nano is everywhere. If you go into the textile also, there are nanofibers, they say that, right? There is a technology which is contributing in the textile development, in a better textile products. There is a that there is a car which was called as nano, in fact, right? And of course it was not in the nanometric range, but the car was small, representing in a small size. So everything, if you see in every facet of technology, things are getting shorter, right? And in the same way, or this is not the proper justification of saying that drug carriers should also be smaller, but we have key advantages which we see in the nano carriers, and dendrimers is one of them. So nanoscience is basically the study of the objects which are measured within nano range. Now, nano, if you talk in the drug delivery, we will come to know that less than 300 to 400 nanometer are ideally suitable for the drug delivery purpose, not beyond that, which we see in the market, or which we actually use for the drug delivery. So naturally also, there are many examples that if you see the adhesive system of GECO, right, which walks on wall, how it remain stick on the walls. If you go into the detail of that, you will find that on the tips of GECO, it has very nanostructured fibers, or you can say the tips are so minute that they have a very good adhesive property, which helps GECO to walk on the walls, right? And so naturally, there is an inspiration of utilizing such a small size for different purposes. And if you see the super adhesive materials which are available in the market, utilizes this technology, or utilizes this inspirational uh, concept of using the nano sized particles for developing the super adhesive material. So it is basically inspired from the nature, that the synthetic nanomaterials which have a strong adhesion power are similar to GECO fingers, right? So you see, nature always gives an inspiration and we as a human being try to mimic nature and apply those concepts, those inspirational mechanisms for the welfare of human being. Now, if you see the range of nanocarriers, I'm sure that many of you already have heard about nanoparticle. I'm sure that many of you have already heard about liposomes. And in the era of COVID, you might have come to know that embisome, amphotericin B, was in demand. And you know that liposomal formulation was very much in the demand during that period. That is also one example of nanocarrier. And there are several others. Like today, we are going to discuss the basics of dendrimers. So if you see in this slide, I have taken some of the very well known, and these are not limited to. There are even more number of nanocarriers other than these also, right? But the major one includes liposomes, nanoparticles, uh, polymeric micelles, dendrimers, right? Mesoporous silica nanoparticles, gold nanoparticles, solid lipid nanoparticle. So many classes of nanoparticles are there. Some of them are, we know that they are in the market. Some of the liposomal formulation are in the market, and the technology is definitely growing day by day. And we will definitely see number of new liposomal formulation in the market, which will be uh, definitely having a very larger impact in the future for the treatment of some very dreadful diseases like cancer. Now, if you see the range of nanocarrier, when when you study in the B form, we realize that the conventional dosage form are of three classes, right? Solid dosage form, liquid dosage form, semi-solid, and even gaseous also, right? You say aerosol is a gaseous one, something like that. But if you see, the most popular one is nothing but the oral, or you can say tablet and capsule. Among tablet and capsule also, tablet is the one which is the most popular one. But when it comes to the rapid action, when it comes to emergency treatment, obviously the parenteral one or injectables becomes more popular. But when it comes to sustained release, for a prolonged release, for a delayed release, for a targeted release, slowly the selection of nanocarrier based system is gaining momentum. And if you see the research output in the last 10 years, many molecules have now have been successfully delivered using different nanocarrier system. And today we are going to understand the basics about dendrimer. I'm not going to include much research data, considering the audience that there might be BPharm students also. So my whole discussion of 1 hour or will be mostly related to the basics of dendrimer. We should understand first what is dendrimer. Like we had studied tablet, what is tablet, then we studied what are the different ingredients or excipients which are there used along with API in the tablet. In the same way, we will try to understand what are dendrimers, what are the types, how we can synthesize, we can prepare, what are the uses, what are the advantages. So basics about dendrimer and how they are applicable in drug delivery as well as targeting, that also we will try to understand. So what are dendrimers? In nutshell, I will say that dendrimers are nothing but polymers. Simply say these are polymers. What kind of polymers? These are hyper branched polymer. I'm sure if you remember uh, the chapter polymer, you might have studied the different classes of polymer. The one best was natural polymer, biodegradable polymer. Another was linear polymer, grafted polymer, branched polymer. This is a polymer which is, of course, branched also, but that branching is a very unique type of branching, which I'll show you in the coming slides. Anybody can interrupt me in between if they have any question. I have no problem with it. They can speak out and I'll try my best to answer. So dendrimers are basically hyper branched. I will what is the meaning of hyper is very high branching. I'll show you multiple points are there where branching you will observe in the structure, and that's why these are called as hyper branched polymeric macromolecular structures, which are monodisperse. They are uh, they exist as a unimolecular structure, and they are highly branched, three-dimensional, and they have three different components which are there in the dendrimer. They starts from the core, they have different layers or branches, and at the last they have terminal functionality. Now this is basically a typical dendrimer structure which you see in this slide, and the core, if you observe in this structure, there is G0, right? That is generation 0. G1 is the first layer, generation first. G2 is second generation. G3 is third generation. And the red dots, blue, yellow, and green dots are nothing but representing the surface functional group, at least in this structure. I'll use the same image to explaining you something different, but can you imagine a structure? For example, I'm giving you an example. Imagine that you have ammonia at the center, NH3, and there are three hydrogens which are emanating from nitrogen, attached from nitrogen, right? And you replace all the three hydrogen with something which is again giving you N, and now N is attached, nitrogen is attached with three nitrogens more, and those nitrogens are again getting attached with new nitrogen molecules, so that again two or more groups can be attached with that nitrogen, and that's how the three branches will grow from the ammonia, and ammonia will called as a core, and the next layer of nitrogen will be, we can call it as a branch or layer or simply generation. Now you have several molecules like this. I can give you example. For example, ethylene diamine, CH2, CH2, and both sides there are NH2. If you replace the two hydrogen of ethylene diamine with acetonitrile, then it will again convert into CH3CN. The CH3CN will get attached to, one of the hydrogen of ethylene diamine. And if you reduce this, it will convert into CH3, CH2, NH2. So again, on one NH2, by replacing one H, we have generated one NH2 again. And again, this NH2 will be attached with acetonitrile, again reduced. And imagine if I am replacing all the hydrogens of the primary amine group of ethylene diamine with such 4 acetonitrile, then reducing those, again new nitrogens will be created, and again we will replace with the two more groups, and that's how it will grow like a tree. In fact, the Greek meaning of dendron is tree. It's a tree-like polymer, right? Start from a core, then comes the layer of branches, and then the terminal functionality. That means the last or the terminal functional group is nothing but we call surface or exterior or terminal functionality, whatever you can say. In between branches, in between layers, we call them branches, and the starting one we call them core. So the core is always called as generation 0. When it is attached with the first layer of branches, we call it as a generation 1, then G2, G3, G4. Now in this discussion, basically I have told you what is dendrimer and what are the different components. And if you read the first slide, you will realize that I have told you what is initiator core. I gave you two examples. The one was ammonia, ethylene diamine. You can use diaminobutane, diaminopropane. People have used even citric acid. They have replaced the OH with the PEG. So actually, this is a tailor-made moiety. You can select an appropriate core which has two emanating groups. You can again make the branches of the terminal groups, and you can actually design, synthesize, develop your own dendrimer. So I'm calling it as a tailor-made molecule, right? It's a tailor-made molecule. Whatever you think you can design, right? Only thing is the chemical structure should be fitting well into the standard definition of dendrimer. So core, interior layers, which I am now giving the new term generations. I have already told you what are these. And the exterior, which is the terminal functionality, and that is called as terminal functional groups. I will show you the real molecule. I have just shown you an indicative structure to make you understand what are dendrimers. Now, this is not only a theoretical molecule. This slide says the ground reality. That means the products based on dendrimers, which are there in the market, and some of them are even at the clinical, trial phase 3, and some of them are already in the market. Some of the well-known products which are there in the market, I have included those in this slide, and you will see that all these products, Viva Gel, Alert Ticket, Stratus CS, Superfact, all these have dendrimers in the core. Basically, these are dendrimeric products, right? I'll tell you what is PAMAM in the coming slides. We are going to discuss a lot, and I'll tell you how this is helpful in drug delivery. Now, I already told you the size, or if I have missed it, the size of dendrimer is usually below 20 nanometer. It is mostly, if you see the fourth generation of dendrimer, mostly the fourth generation of PAMAM dendrimer, it is around 15 to 20 nanometer. If you purchase that from the Sigma, the standard one, which is available in the market. And now you must be understanding, when I'm saying the fourth generation of PAMAM dendrimer, you must have realized that core is there. And with the core, there are 4 different layers of the branches. And that is called as fourth generation of dendrimer. So the size range of the dendrimers is around 20 to 30 nanometer in a standard way. And this I'm talking about the basic dendrimer. Now, what are the advantages? If you count those, I have mentioned some of those. I told you the one which is size, and how the size is advantageous. I gave you one example which is not related to drug delivery, but we will come up with this in the future. But I told you how super adhesive materials, if it is being prepared using nanomaterials, how they are really behaving a very good or super adhesive material. So nano size is having its own impact in drug delivery, and it is advantageous. One thing I told you, that at the surface, I gave you one example of NH2. There can be OH termination also. And again, we can attach with the surface different targeting group, different drugs, right? It has a very high drug loading capacity. Drug can be encapsulated inside dendrimer. I'll tell you how, and how it is very much helpful, how it resembles micelles, right? And how it can solubilize the hydrophobic drugs. And it has very little cytotoxicity. And this cytotoxicity is one of the disadvantages also. When you will come in the future discussion, you will realize that there are many NH2 groups which are there, and which leads to cationic charge or cationicity in the carrier system. And that's how these becomes challenging to deal with, or we can say that it is highly toxic to RBCs. Now, at one point, the cationicity is showing the toxic results, but at the same time, it is helpful in the membrane transport also. So you will see that they have an excellent membrane transport capacity. In fact, for the transfection studies, dendrimers is one of the gold standards, apart from lipofectamine and other membrane transport agents, and dendrimer is one of them. So these are the key advantages which we will also discuss one by one and will come up. Now, if you see the standard synthetic methodology to synthesize dendrimer, either you can start from the core, or either you can start from the terminal point. So if you start from the core, I gave you example of ammonia and ethylene diamine, and you might have realized that when ethylene diamine is attached with acetonitrile, and then acetonitrile again we reduce it, and again we get a new NH2. In the same way, if you see the divergent approach in this slide, which is starting from core, the first branch get attached with all the four molecules of the core, or simply four atoms of the core, I should say, and then again two new connecting points are being created. So there were 4 points of attachment, and then in the first generation 8, and after that 16, and then this 16 became 32. So we are doubling the connecting points, and that's how the generation is increasing. That is what we are calling it as a divergent approach. It is getting diverged, right? And if you start from the terminal functionality, the last point, and if you go inside, so we are converging to the center. And that's why this is called as convergent approach. The very standard methodology as far as chemistry is concerned: divergent synthesis, convergent synthesis, starting from the terminal functionality towards core, and from core to the terminal functionality, we call it as a divergent. Two synthetic methodology, or simply method of preparation for dendrimer. I'm again taking the same structure to make you understand why we consider this carrier system for drugs, why we consider this carrier system for delivering even genes, and why we call this as a versatile carrier. Something we say versatile means having multiple capacities, can be utilized for multiple purposes, right? Can be used for drug delivery, it can be used for targeting, it can be used for gene delivery, it can be used for solubilization, it can be used for you know uh, kind of dual delivery or multiple drug delivery, even for RNA, DNA delivery. I'm talking about only one carrier which can be utilized for these much different type of applications. So now you realize that in this figure, the red dot, if you imagine that it is a PEG, we can attach polyethylene glycol on this surface, because this is a real polymer, real chemical molecule. It's not an assembly, it's not a micelle, it's not a fabricated molecule. It's a polymer. It's a real polymer having a covalent bond attachment with the core and branches. And at the terminal surface, you can attach PEG or any ligand, right, which can help in targeting of drugs. You can even conjugate drug, you can encapsulate drug. I will tell you how. Even you can change the surface. You don't want NH2 terminated dendrimer. We have OH terminated dendrimer in the market. We have COH terminated dendrimers in the market. We can synthesize something new which is not having any core or branch of synthetic origin. We can simply synthesize a natural dendrimer. You can utilize any biodegradable molecule like citric acid, and you can attach number of PEG molecules. It will become a completely biodegradable dendrimer. You can convert lysine into poly L lysine dendrimer. Lysine is basically an amino acid. It is a completely biodegradable dendrimer. So that's why I told you tailor-made properties. One, versatility. You can attach many things at the surface if you wish to, as per your requirement of drug delivery. If you want to attach an antibody, yes, you can attach it. If you want to attach a ligand, for example, folic acid for cancer targeting, yes, you can conjugate it. You can attach PEG. You can even conjugate certain drugs, right? And so many other things are possible as far as the surface of the dendrimers is concerned. And that's why this carrier system is considered as a versatile carrier system. Now, I'll show you how the drug is encapsulated inside dendrimer. Before going into that, I'll take you to the real dendrimer structure. And this is the one class which I'm telling you is one of the most popular class of the dendrimer, and we call it as polyamidoamine dendrimers, PAMAM dendrimers in the short, right? If you see into the detail of this structure, you will realize that in the core there is an ethylene diamine. You see CH2, CH2, and then N, and at the other terminal also N. That N has been attached with two new NH, and then again that N is attached to another N, and then so on. And if you see, the branches are getting enhanced, and one by one this is getting larger, larger and larger, but its size remains within 20 nanometer. And that's how this structure, which is there in front of you in this slide, is fourth generation of palm dendrimer. Right, you can see easily the bonds at the terminal. You can see the number of NH2 groups are there. And now, how drug can be encapsulated? If you see the inside of this structure, there are void spaces. See, I'm moving my cursor, and you can see that there are void spaces where drug can come and can be encapsulated. How? You see, there is an NH in the inside structure, right? And suppose there is a drug having O or oxygen at the surface, or it can be used to make an ionic bond between the tertiary nitrogen or secondary nitrogen, as well as hydrogen bonds between the present NH and the oxygen present in the drug. And that's how drug will remain attached inside, and we can say that drug can be encapsulated. And I'll show you certain more figures in the coming slide. See, if you consider this as a dendrimer basic structure, right, the orange oblong structures you can consider as a drug, right? And at the surface also, the green one, you can see that this drug can also be conjugated at the surface. It can be encapsulated physically with ionic bond, with hydrogen bond, with van der Waals attractions, and it can also be conjugated at the surface. Right, it can be adsorbed at the surface. Suppose NH2 is there at the surface, it will convert into NH3 plus, and any drug having a negatively charged surface, it can make an ionic association, ionic bond, can remain attached at the surface. It can be adsorbed also sometimes. And that's how these are the different mechanisms which you will see that dendrimers can use actually to encapsulate drug. So when I ask you a question that how drugs can be encapsulated inside dendrimer, the best example I told you is by van der Waals, by hydrogen bonding, by ionic bonding, even by covalent conjugation also. The different electrostatic interactions leads to encapsulation of drug inside dendrimer. Right? Now the inside cavities of the dendrimer, for example, this is also another class of dendrimer, which is called as polypropylene imine dendrimer, and it is nothing but we call it as a PPI dendrimer in short. One class was polyamido dendrimer, another I'm telling you is polypropylene amine. You can see in the core of this PPI dendrimer, the core is diaminobutane. You can see one CH2, 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 four CH2 groups are there, and at the terminal, two nitrogens are there. Diaminobutane. Then again, that nitrogen got attached with another, nitrogen, and then again, such the branch was increased, right? And this is how the polymerization takes place. And you can see the void space, right? You can see the vacant space inside the dendrimer, and at the top, at the surface, I can say the exterior, there are n number of NH2 groups. So many NH2 groups are there. When amine groups are there, primary amine groups are there, they are going to be absolutely soluble in water. But see the inside architecture of this structure, it is tertiary nitrogen. This is completely hydrophobic area. Can you remember one carrier? Can you remember one very good structure of micelle? If you remember, micelle were made up of hydrophilic and lipophilic molecules, right? And there was an hydrophile, there was an lipophile. And when you keep on adding the surfactant in a water, what happens? When they reach a critical micellar concentration, they assemble themselves in such a way that the hydrophilic heads remain in contact with the water, while the hydrophobic tails remain at the center or at the core, where drug can be solubilized. You might have remembered that Drug solubilization can be achieved using surfactant with the formation of micelle. Here also, you are actually observing almost the similar structure, but the micelle was an assembly of the hydrophilic lipophilic molecules or surfactant molecule, but this is a real molecule. This is a covalent bonded molecule, or you can say a polymer, which cannot be disassembled, right? It will not, if you dilute it, it is not going to rupture. When we dilute our micelles below CMC, the micelle will rupture. The drug will leads to burst release. But here, the drug is there inside a polymer, which is behaving like a micelle, which looks like a micelle, but it is called as unimolecular micelle. It is a real molecule, it is a polymer, right? And it cannot be disassembled just by dilution. So, what was the biggest challenge with the micelle? When we solubilize a drug using surfactant by formation of micelle, if those are injected into the blood, suddenly they are being diluted with the 6 liter of the blood in the body and leads to reduction in the critical micellar concentration of surfactant, leading to burst release, which is not possible in this type of carrier system. So you might have realized that dendrimers are hydrophobic from inside and they are highly hydrophilic at the surface. And these are the excellent solubilizers. That is why these are able to solubilize n number of drugs. Insoluble drugs are very well solubilized using dendrimers, right? So this is one of the very good advantage, that how drug can be encapsulated inside dendrimers. Now, without taking much time, I'm not discussing about the key properties of palm and PPI. I'll tell you, palm is one of the most popular, but PPI is also not behind. It has its own advantages of being more hydrophobic than palm, but these are the two major classes. There are many more which I have not taken in this discussion, but like polylysine dendrimer, citric acid dendrimer, uh, there are triazine dendrimers which are there, melamine dendrimers are there. So lot of classes of dendrimer are there, but the most studied, and if you see in the slide which I have shown you in the market products also, you see there are only mostly the palm dendrimers which has been explored for drug delivery because of its key advantages. Now, as I told you that these are excellent penetration enhancer also. So this cationic moiety or the cationic structure, if you see, the A part of this slide is nothing but a cell membrane. And when this cationic molecule, which is dendrimer, now why it is cationic, I told you, because number of NH2 groups are there. When primary amine is there, definitely the surface charge is going to be cation. So when it comes in contact with the cell membrane, it get adhered at the cell membrane because the cell membrane is having anionic charge at the surface. So it has an excellent addition of cationic and anionic charge, and that's how dendrimer get internalized. Transfact, which we say that, because it crosses the membrane and because of the charge it enters, as well as because of its very small size in the interstitial spaces also, and it leads to entry inside the cell. Why this is a very promising factor? Because if you want to deliver DNA inside cell, if you want to deliver gene inside cell, dendrimers can be an excellent carrier. I'll show you in the coming slide. See, this is one slide I have taken from one of our review articles, that why DNA get attached with the dendrimer, because dendrimer is cationic in the surface, right? DNA is anionic at the surface. They mix, both of them makes an excellent complex, and dendrimer and DNA complex is called as dendriplex, right? And this dendriplex is having the capacity to get entry into the cell. This is highly helpful in delivering DNA, RNA inside cancerous cell, and that's how we say that dendrimers are excellent carrier for DNA, RNA. This is the same thing. Dendrimer are cationic, DNA is anionic. Obviously, it will make complex, and this is the basis of gene or DNA delivery, right? So the basis of gene and DNA as I told you, if you go into the size of the dendrimers, you will realize that the size of the dendrimers is very small compared to long chain of DNA. And you see how DNA remains bind with the dendrimers. You must have remembered the histone protein structure of DNA. Histone, H1A, H1B, if you remember, histone and non-histone proteins of DNA. In the similar way, DNA get attached, make a complex with the dendrimer, and overall charge remains to be cationic, which helps in the delivery of DNA to the cell. Now, I already told you why it is helping in drug delivery. And you see, I told you that there are primary amines at the surface, there are tertiary amine at the surface, and that's how different pH also exists at the surface as well as interior of the dendrimer. And not exist, but different protonation states exist at the tertiary end or at the inside or at the exterior. And when we change the pH, it is observed that these amines can be protonated, can be deprotonated, and based on that, we can encapsulate it. So surface charge plays a very key. And in this structure, I can tell you how they are helpful in targeting. Now, when it is an acidic environment, the tertiary amine will remain protonated, right? As well as primary amine will also remain protonated. That means they will have a high cationic charge. Anything which is encapsulated inside dendrimer in the acidic media will lead to repulsion between nitrogen, and hence the drug will be released. And if you see the physiology of tumor have a physiology, or the tumorous site have a pH which is less than 7. Less pH is representing the acidic nature. So whenever the injected dendrimer with the anti-cancer drug passes through the tumor, it will be exposed to the acidic environment, and in the acidic environment it will get protonated, and with the protonation the branches will repel each other, and that's how the encapsulated drug will be released automatically in the tumorous area. And that's how we will be able to target the drug into the tumorous area directly based on simply the structure and properties of the dendritic. So acid labile release of the drug or pH based tumor targeting of the drug can be easily achieved with the dendrimers. I gave you one example where no ligand was there. Based on its structure conformity, dendrimers were able to release the drug at the tumorous site. So this is one basis of targeting. We are slowly heading toward targeting. I told you how drug can be delivered, how can be solubilized, how drug can be solubilized, why gene delivery is possible. The dendrimer, and a lot of studies and products are there now which are based on dendrimer based gene delivery. But I have till now told you only the advantageous part of the dendrimer. What are the challenges? What are the disadvantages? This cationic charge itself is a disadvantage. You see, if it is a cationic polymer injected in our body, injected into the blood, this leads to hemolysis. It ruptures the RBCs. So there are many studies. You see the fourth generation, the second row of this table, which is there in this slide. You see that 35.7% hemolysis was reported once using fourth generation PPI dendrimer. That means such an hemolytic molecule dendrimer is. Now what is the solution? The solution is we should convert this dendrimer, or we should prepare the dendrimer using biodegradable core, biodegradable branches. For example, if we make the dendrimer with polylysine or L-lysine, you see the fourth generation of polylysine dendrimer is having 14% of hemolysis, which is very less than comparison to fourth generation of PPI and PAMA, right? Another is coat, or we can conjugate the surface of dendrimers with certain molecules like PEG. We can coat the surface of the dendrimers with certain sugars like mannose, even other carbohydrates, which will hide the cationic charge, and hence the hemolysis can be reduced. You see, there are many studies in which glycine was coated, mannose was coated, lactose was coated, PEG was coated, and that led to reduction in the uh, toxicity associated with the cationic dendrimers. So this is one way that by surface engineering, by attaching something more at the surface like carbohydrate, which is biodegradable and which hides or which reduces the cationic charge at the surface due to amines, we can achieve the reduced hemolysis. So this is something advancement in the carrier, and see that dendrimer surface has been conjugated with so many properties. This is one structure or figure in which I have included various molecules which were attached at the surface of dendrimer like mannose, galactose, lactose, leucine, tyrosine, alanine, tryptophan, arginine, lysine, glycine, PG, etc., etc. Sialic acid, and many more, folic acid, even antibodies, dexamethasone kind of drugs, right? So many, even drug, ligand, antibody, lot of things has been attached at the surface of dendrimer for different purposes, targeting maybe biodegradability, maybe solubility, maybe antigen antibody interaction. So many purposes are there which can be utilized, which can be explored by tailor me by surface engineering the surface of dendrimers. So this is one. I'm not discussing dendritic box. It will be more to add core research, but it is something like sealed dendrimers can also be prepared opposite to what we are discussing. Now pegylation is nothing but attachment of polyethylene glycol at the surface of dendrimer. It has been observed that pegylated dendrimers are more advantageous even than the pure dendrimers. So people have done in the past pegylation, different degree of pegylation. That means partial NH2 were conjugated to PEG, partial were conjugated to no molecules, or no molecule was conjugated to half of them, and that's how pegylated dendrimers were prepared. They have their own advantages because it improves the solubilization and encapsulation further. So many drugs like 5-FU and many more like have methotrexate, cisplatin, docetaxel, paclitaxel has been successfully encapsulated. Higher drug loading was observed with PEG compared to pure dendrimer, and that's how we see that higher drug entrapment can also be achieved using pegylated dendrimer. So these are certain examples I'm giving you that why not only basic dendrimer is advantageous, but the pegylated one can also be even better. Now, apart from drug delivery, gene delivery, solubilization, targeting, there is one more application which I have not discussed, and not much I'm going to discuss at this part, but I want to make you aware, that is diagnosis, right? So it is a very well carrier used for magnetic resonance imaging nowadays in the boron capture therapy, boron neutron nucleus capture therapy, and it has its own applications as far as diagnosis is concerned. So you will see that a lot of research is ongoing which reports the application of dendrimers for the purpose of diagnosis of different diseases. So in one carrier, you might have witnessed by now that solubilization, gene delivery, drug delivery, targeting, diagnosis, these n number of applications are there. And I told you, with the help of hydrogen bonding, hydrophobic interaction, ionic interaction, we can change the different generation of dendrimer with different surface, and we can get the solubility enhancement of different drugs. I have included this table from one of our published review article, which reports the number of drugs which has been successfully solubilized using dendrimers. You see paclitaxel, flurbiprofen, indomethacin, propranolol, right, diclofenac, pyrene, mefenamic acid, naproxen, niclosamide, ibuprofen, ketoprofen, right, nicotinic acid, artemether, these all and even more. This is an old table, I can say, but you will see that more than 100 drugs has been successfully encapsulated, solubilized using dendrimers. I told you why it is a solubilizer. I already told you my cell comparison with the dendrimer. And apart from this, you see that this carrier can be delivered using different routes. IV, IM, transdermal, ophthalmic, these are the major routes people have explored for delivering dendrimers. And we have seen that it has reported in a very successful, results. And this is again related to, cell uptake. I told you how this can be utilized for taking drugs inside the cell. A fluorescent dye was attached, and this is how we can study the cell uptake using dendrimers. So, sustained release, controlled release. I have not discussed this much part, but yes, it is a sustained release carrier system. It slowly releases the drug which is encapsulated inside dendrimer, and that's how we call it as sustained release carrier. Of course, it contributes into this equally, and that's how it is a very useful carrier system. Now, I tell you that there are different ligands which can be utilized for targeting drugs. For example, mannose was conjugated to the surface of PPI dendrimer in this structure, and you can see that the alveolar macrophage uptake of rifampicin was increased very high when it was delivered using mannose-silated, that is mannose conjugated to dendrimers, and that's how the alveolar macrophage uptake was very high for mannose-silated dendrimer. So you can attach targeting ligand. I told you these are certain examples which I have taken, but these are not limited to. And if you do one by one, these are different studies which reports the targeting potential of dendrimer. So I'm reaching to the end of my discussion or my delivery of today's topic, and I can tell you that this is a very versatile carrier. And I tried my best to make you understand that what are dendrimers, what are the types, how we can synthesize, what are the advantages, some of the key disadvantages, why it is versatile, how it can encapsulate drug, how it can solubilize drug, how the surface can be engineered for targeting for the purpose of antibody antigen reactions, even for transfection, for studying cellular uptake. So different aspect of dendrimer I have tried my best to tell you and to discuss you. And it can be delivered using different route. It can be administered using different routes, which proves its versatility. With this, I would like to thank my team who helps me to do this type of research. I have 4 research scholars in my team right now, and 4 master's students. One is Sarita, another Rakesh, Vinay, and Sanya, who are my team member, who we together work as a team, and we try our best to give some output in the form of publication, presentations, as well as other research work. Thank you so much for your patience listening. I hope I could deliver the basics about dendrimer, and I hope in the audience you were able to understand what I did today. Thank you so much.
Thank you very much, sir. Thank you very much for this wonderful talk on dendrimers. And sir, I have received a lot of questions also
Sure.
from the participants that I would like to put before you. And the very first question is that how it is better as compared to micelles, so far as drug delivery is concerned.
Okay. So as I told you that micelles are basically assembly of surfactant molecule, right? micelle is not a real molecule. If you go diluting micelles, the micelle will rupture, right? We always say that micelles are formed above critical micellar concentration. So that means if we dilute them, again they will get separated out as a hydrophilic, lipophilic, or simply surfactant molecule. Now imagine that you have solubilized a drug using a micelle, and that is now injected into the blood of a human being. Now, as soon as it is injected into the blood, it is getting exposed into 6 to 7 liter of blood of a particular individual. That means it is getting diluted. And if this dilution is there, it will lead to burst release of the drug. Imagine the case of a dendrimer encapsulated drug. Now, it is not an assembly, right? You understand? I hope you understand what is the meaning of assembly and what is the meaning of covalently conjugated, bonded molecule. So when you encapsulate drug in the molecule like dendrimer, and when it is injected also, even if it is diluted 10,000 times, that molecule is going to remain as a unimolecular structure. So though it is similar to micelle, but the assembly will not disrupt. It will not lead to burst release. That is one of the biggest advantage or comparison you can, tell about micelle.
And the second question is that once we go for pegylation, then all the surface moieties will be replaced by the polyethylene lipopyl molecules. So the only option
Right.
we left up with is just putting our drug into the hydrophobic core of the dendrimer, sir. So in that case,
Okay.
would not be the drug, there would not be a leakage problem when we'll be administering the drug.
Okay, I can tell you in this way. First thing, don't think that all the NH2 molecule will get conjugated to PEG. Suppose there are 64 NH2 groups and I am only allowing to react half molar concentration of PEG with this dendrimer. That means some of the NH2 will remain as such, right? Not all the molecules of NH2 are going to be occupied by PEG if I am doing a controlled conjugation. Okay, controlled means if there are 64 NH2 available, I am going to only react with 16 molecule of PEG. Molar ratio I am keeping less. Now leakage problem will be very less. Why? Because at the surface there will be convergence of the surface molecule groups. For example, PEG is having a very long chain. Now when it will be bonded at the surface, it will also get fold at the surface, which will lead to shielding of the surface, or you can say sealing of the surface. So yes, there will be some percentage of leakage, but the majority of the drug will remain encapsulated because there is hydrogen bonding or van der Waals attraction or ionic interaction which exists there inside the dendrimer, which is keeping it inside, and that's why it will be released in a slow manner. So yes, the answer is leakage will be there, but there will be less leakage if you compare with the other carrier system of the similar, structure.
And I would like to take up one another good question. That is, what is the possibility of delivering proteins with this carrier?
Okay, see, with the protein, insulin, people have tried to deliver using dendrimer. Only thing is, it is a cationic molecule, but there are anionic dendrimers also available in the market. Yes, this can be utilized for delivering proteins. But when it comes to conjugating any protein or making a chemical bond, you need to take care of selecting the different, I should say, carriers, different solvents, different reagents, which should not harm the integrity of the proteins which you are intending to deliver or you want to deliver using dendrimers. But yes, it can be utilized for delivering proteins. It has already been explored for DNA at a large scale. You can see in the literature. Yes, protein it can also be used for delivery.
Okay. So, thank you very much. And I do not have any further questions. So,
Okay,
thank
sir.
you, sir. And we have with us Dr. Lalit Lirajam, madam, Principal School of Pharmacy.
Hello.
So, I
Namaste, madam.
request him to say.
Namaste, sir. Good afternoon.
Namaste, madam.
Thank you very much, sir. Very nicely you have taken the session. Thank you very much.
Okay.
In fact, whenever you have time, please visit our university also.
Sure,
We
madam.
can,
Sure.
yes, sir. We can have offline expert also.
Sure, sure.
Yes. Thank you so much, sir.
Thank you so much for giving me opportunity to share some of the concept related to Dendrimer. Thank you so much.
Sir, take a moment. Sir, I would like to present this certificate as a token of appreciation from Parul University family to you. So kindly accept the certificate, sir.
Thank you, sir.
And sir, it was in fact very good to hear from you. It was quite interesting. So thank you very much for a elaborative speak on Dendrimers, and starting from the basics of Nano, you
Yes,
touched upon every aspects of the Dendrimer. That is their
yes,
introduction, current status, various generations, and possibilities of drug delivery and targeting. So thank you very much, sir. Thank you very much once again,
thank you, sir.
and would like to hear from you again and again in the future also.
Thank you, sir.
Thanks for spending time. Thank you.
Thank you so much.
And for all the participants, you shared the feedback link on WhatsApp groups, and also we are going to share it on Facebook. So you are required to kindly go through it and kindly fill the feedback form. Thank you very much.